Conservation of promoter function in evolution depends on the conservation of the corresponding promoter structures. Therefore, an assessment of the evolutionary conservation can help you decide which of the dozens of binding sites that you typically find in a promoter are most likely to be functional. In this tutorial, you will learn how to retrieve orthologous promoters and identify conserved binding sites in the alignment of their sequences. We will be looking at the promoter of the human IL-10 gene. Interleukin-10 is a cytokine involved in immunoregulation and inflammation. We start with a search for the gene in our genome annotation Eldorado and will continue with the alignment program Dialign TF. To search Eldorado, you just select the organism, human in this case, and enter a gene identifier. I used the gene symbol here, IL-10. Then you can run the search. We get to the Eldorado overview page for IL-10, where we can select different views of the annotation for this gene. For the IL-10 promoters and their orthologs, we need the Comparative Genomics view. This view gives us a list of the orthologs of IL-10 in different vertebrates, with their promoters and transcripts. Orthologous promoters are grouped into sets. The two promoters in the human IL-10 gene belong to sets 1 and 2. Let's run a comparison on set 1. The promoter sets can be selected in this list. So I select set 1 here. We have a number of options for running a comparative analysis on our selected promoters. With Dialign TF, we'll get a multiple sequence alignment that also shows us any transcription factor binding sites that are conserved in aligned regions. I'll just change the number of nucleotides per line in the output here. Everything else stays at the default values. We'll be looking only at very conserved transcription factor binding sites that are present in at least 11 out of 12 orthologs and need to be in aligned regions as well, which means they are at equivalent positions in the promoters. In the alignment, we see the different sequences from rhesus monkey to platypus, the alignment position, the sequence conservation, and in color, the transcription factor binding sites. Only two different sites, $stat for the stat factor binding sites and $VTBP for the vertebrate Tatabox motif, are sufficiently conserved to be listed here. The transcription start sites will be shown in red. The conserved binding sites are just a bit upstream of the transcription start sites. The stat sites are blue and the Tatabox is purple. The sequence adjacent to the stat site is not very well conserved. We see unaligned sequences in mouse, dog, cow, and platypus, so the diagonal similarity of the sequence at the site itself gets only 4 out of 10 possible asterisks. Conservation of a stat site in the IL-10 promoter also makes sense biologically. STAT3, for example, is a known transcriptional regulator of IL-10. If you want to learn more about this and other functional relationships of IL-10, consult the More Gene Info page on the Eldorado overview output. More Gene Info gives you comprehensive information on the function of a gene. Here in the Interaction section, we find literature references on the interactions of IL-10 with other genes and proteins, with direct access to the relevant PubMed abstracts. For example, this publication on activation of IL-10 by STAT3. That's it. Thank you for watching this tutorial and, as always, don't forget to have a look at the other tutorials on our site.